Hello, everyone. Welcome to the review session. In a moment, I'm going to share the presentation with you all. Gotta do a quick system check, make sure I can see everything. So you should now see a link at the top of the screen, which will allow you to ask questions during the presentation. We'll let some more people join, and then we'll get started. I'd noticed there's not many questions that have been asked. So if you have questions, I would recommend using the question link at the top of the presentation. If someone could test that out for me, it'd be greatly appreciated. And we'll get going here in a few minutes. Whoever Xander Schwartz is, thank you for testing that out for me. I appreciate that. See, we have 17 viewers. Yes, you understand everything, Gavin. I can see your question. So when questions pop up, feel free to ask. Hi, Daniel. If you have questions, I'd recommend you use the presenter view, the link that was posted. All right, it is 8:05. I can see we have 18 viewers. I'm assuming you all can hear me just fine. At least I hope you can. We're going to go ahead and get started here. So semester one review. A couple things. So if you have questions, please ask them. I can see some questions are already popping up in the question and answer link. So please use those questions. I'll try to answer them as fast as I can. I might not see your question pop up right away. Let's get going. So someone asked one question about the purpose of gel electrophoresis, so we'll definitely get into that. But I want to first kind of just talk about some of the labs we did. So if you look at these labs, we have several labs in this first unit that kind of set us up for the whole semester in terms of techniques and skills that we were trying to teach you. So this termite lab was the very first lab, really taught us two major things. Taught us the difference between qualitative and quantitative data. So you're gonna have to be able to not only identify them, but be able to apply your understanding of them on the final. You got to know the difference between qualitative being more descriptive data that can be observed but really not measured. So examples are colors, texture, smells, taste, appearance. Think of quality when you think of qualitative. That definitely helps you remember it. And then quantitative is dealing with numbers and measurements, things that you can measure like length, height, area, volume, temperature, sound, cost, number of people in a group, those kinds of things. So quantitative means quantity. That's where those words come from. So if you look at this picture here, you have two examples of an oil painting. The qualitative data would be the color of it, the smell of it, the texture of it, the description of it. The quantitative data would be how big it is in inches or centimeters if you're really doing it in science class. The weight of it, surface area, those kinds of things will all be quantitative data. And then we also learned a little bit about independent versus dependent variables. Those are very important to be able to identify. The best way to think about it is that the independent variable is the variable that the researcher, the experimenter, is changing. It's the action being taken place. It's what you are doing in the lab. It's what you are manipulating. And so in this example, you have adding fertilizer to a plant. That would be the independent variable. That is what the researcher is testing. They're testing to see the effects of fertilizer on plant growth. Dependent variable is what is affected or what is the outcome, what is changed by the independent variable. So we have different things we could collect. We could measure plant growth. We could look at number of leaves produced. We could look at if the plant produces seeds or anything that could be measured or observed. It could be qualitative data based on what the fertilizer does. So the dependent variable tells us if our independent variable is having an effect or doing anything in our experiment. So those are the two major things from that termite lab, was identifying independent versus dependent variable and really understanding the difference between quantitative and qualitative data. One real important thing about the independent variable is you've got to always remember that good experiments have only one, because you got to know if, for example, this fertilizer is causing a change in plant growth. If you had been adding different amounts of fertilizer, different amounts of water, giving the plants different amounts of sunlight, all three of those variables could have an effect on plant growth. You don't know which one is actually impacting the outcome of the experiment. So a good experimental design has only one independent variable. Everything else is held constant, kept the same. We call those controls. Those controlled variables could change, but we keep them the same, so we only can manipulate one, the independent variable. All right, here's a great question someone brought up. Can't colors be measured by a I think you meant spectrophotometer. Yes, that's true. And if we do measure color through spectrophotometry, that would be a quantitative measurement. In fact, that was one of the labs we did. We measured color using spectrophotometry. I don't have a picture of that. Let me grab a picture real quick and dump that into the slide deck and talk about that. So spectrophotometry definitely is a quantitative measurement. We did that in the Lance Armstrong lab where we measured how blue the substance was. Let me go ahead and dump that in real quick before we move on. I'll definitely talk about gel electrophoresis as well, but we might as well talk about spectrophotometry and Bradford solution. Bradford. Image. All right, let's jump back in. Oh, any other questions about independent, dependent variables, quantitative, qualitative data, anything like that? I see a lot of questions pouring in about protein synthesis. We'll definitely get to that. And then somebody asked, describe the purpose of gel electrophoresis. We're going to talk about that next. So let's go ahead and move on to that topic. So that was our candy dye lab. Gel electrophoresis was the candy dye lab. So tRNA lab was independent, dependent variable, quantitative, qualitative data. Candy dye lab was gel electrophoresis. What's going on in a gel? So let's talk about gel electrophoresis. So first of all, you should be able to look at a gel and understand the principles of what are going on here. So I have a gel picture here. I think it's straight from the study guide that shows us a lot of information. First of all, these bands represent molecules. So a band on the gel represents a certain molecule. So let's call this A, B, C, D, E, and F. They could be DNA molecules. They could be proteins. And the whole purpose of gel electrophoresis, to answer this question, this is what is the purpose of gel electrophoresis, is to separate molecules based on their size. And there's some principles you need to know. You need to know that the bigger molecules travel slower. So like this red one right here didn't go as far because it had a hard time moving through the gel. And so the bigger molecules, the bigger in size, I think about DNA, it's number of nucleotides long. Think about protein, number of amino acids that make up that protein. The size of the molecule determines how far down the gel the molecule will move. And so from this gel, we could interpret that C must be the smallest molecule present and A must be the largest molecule present. We could also interpret, and this is a minor detail, that A, B, C, D, and E all have a negative charge because they seem to be moving towards the positive end of the gel, where band F, molecule F, represents a molecule that's a positive charge. It went backwards because the charge is what moves the molecule through the gel. If the molecule had no charge, it would stay in the wells that we pipette them in because there's nothing attracting it or pushing it or pulling it to one of the sides. So big molecules move slow, small molecules move fast. Now, the thickness of the band gives us a more of a qualitative idea of how many molecules or a quantity of molecules, how much we have in each sample. So we could basically predict that we have more molecules in sample A than we do in sample D. Doesn't mean A is bigger, it just means we have more of them. Where it doesn't mean D is any smaller, it just means we have, or I mean D is smaller, we just have less of them in terms of the thickness of the band. So thickness gives us a qualitative observation or a qualitative piece of information of how many molecules, how much we have. So another thing we learned about the gel, and this came a little bit later on in the year, but it's going to be a practice, or it's a skill you need to know for the final, is we can measure how far these bands migrate, how far they move, and we can plot them on a graph and use a graph like this to figure out the size of an unknown piece of, for example, DNA. Let's say this molecule moved 25 centimeters down the gel. Well, we go find on our graph here where 25 is, so 25 is right around there, and we follow that all the way up to where it hits our best fit line. So here's where 25 meets the best fit line, and then we follow it over to the y-axis and figure out, well, it's roughly 500 base pairs or whatever the unit is big. And so that's called interpolating data. We did this a lot this semester, so you should be able to do that skill, is interpolate what's going on in a gel. So those are some gel electrophoresis questions. Let me take a look and see if there's any other questions on gel electrophoresis before we move on. All right, so I believe I answered this question. What does the thickness of a band in a gel lab tell us? It tells us that there's relatively more or less of the molecule present, but it does not tell us how big it is. And that's probably the biggest mistake people make, is that they think the thickness of the band tells us how big it is. It does not. It tells us how much we have. Any other questions on gel electrophoresis before we move on? All right, we seem pretty good on that. So let us move on to the next slide and talk about spectrophotometry. So spectrophotometry helped us figure out how much molecule there was in a sample. So we look at a spec. The spec basically tells us, it's measuring how much light is being absorbed. So I go back to this picture here. Let me pull up my laser pointer. When we shine, here's our sample. So think back to the Lance Armstrong lab where we had different color blues in our sample. And that blue came from Bradford solution that turned blue in the presence of protein. So we wanted to measure how blue our samples were, because we made the connection that the more blue there it was, the more protein found in that sample. And so what we did is we shined light on it. We learned that light either can be absorbed by molecules or, in this case, reflected. Or in this case, it actually shines through the sample. So the more light that it's absorbed, the less light that gets to this detector, and the machine determines how much light was actually absorbed by the sample. So the higher this number, the more blue or the more molecules there were in our solution. And we use that principle to figure out, if we have absorbance here, we can figure out, if we know that the absorbance is 0.8, we can go over on this line, drop it down, and figure out how much protein is found in that sample. So, if we look at 8, it'd probably be somewhere around, let me look here, probably somewhere around 100 or so. I think 8 hits right, 0.8 hits right there. So same idea, though. We can use interpolation of data points. We can look at a sample and go, if it's a 1, follow it over, drop it down, find the actual protein concentration in that sample. That was our spectrophotometry lab. Okay, so this question here, what's the purpose of the spec lab and PCR labs? Okay, so let me go find a picture of the PCR lab for me to drop in that presentation. Actually, I think there's a good one in the study guide, to tell you the truth. Here we go. This picture right here, let's see if I can copy and paste that into the presentation. Nice and big. So PCR labs, to answer that person's question about the PCR lab. If you look at this image and we look at the difference between band A and band B is much thicker. Band A is much thinner. So this is an example of a PCR sample of DNA. And so the more cycles the sample goes to, the more copies we make, the thicker the band is. So PCR basically is a tool scientists use to make copies of DNA. We're not going to really get into the details of how the copies are made, but it's just copying that molecule. So the more copies you have, the thicker the band. So PCR, one practical purpose of PCR is when you find a sample of DNA, in order to see it on a gel like this, you don't want really scrawny, skinny bands that are hard to see, especially if you have like one copy of DNA, you won't even see it, which is what lane 1 here, or lane 2 is showing. But if you have lots of copies, it's much more easily viewed. And so samples are usually PCR'd before they're run into a gel, just so we can visibly determine how big the molecule is based on the DNA ladder on this side. Another purpose of PCR we did in our GMO lab was actually to try to find if a certain gene is found in an organism, where we use these things called primers that find specific codes, and then if the codes match up, the PCR process makes copies of that gene, and if the band shows up, it tells us, hey, there's the gene is present. So PCR can help us identify genes. But again, for the final exam, I would not really concern yourself with how PCR works. Just kind of know the end result of PCR is more copies of DNA. All right, this question asked about SDSPAGE. So we'll go over that. That wasn't really in the first unit for the bio curriculum, but we did cover it in our protein. We talked about proteins, so I might as well find SDSPAGE gels to show you. As far as what SDSPAGE is, you don't really have to know. Here's a good picture. I like this picture. I've seen this one before. You don't really have to know how SDS works. What you do need to know, though, is basically the same principles of gel electrophoresis. That SDSPAGE is just another way of seeing how big, in this case, proteins are. And what I like about this picture is that it really gives you a good idea that this is the gel. The gel has pores in it, and the bigger things get stuck in the gel, whereas the smaller things down here make their way out faster. And so it just sorts out proteins. Same concept, though. The big ones travel slow, the small ones travel fast down the gel. Okay, it looks like that's all the questions from the first unit. Unless there are any more. We're going to move on. So far, no one's posted anything here, but let's just talk about the central dogma. So this is probably one of the cornerstones of our semester. This concept, the idea that DNA is this molecule found in all living things. All living things on this planet contain DNA. And DNA is used to make proteins. So the central dogma of biology, which means all living things go through this process. All living things have DNA, and in some fashion, they take that DNA and they transcribe it, and then they make RNA from it. And then they take that RNA molecule, and from that, create proteins. And proteins are the real functional molecules. They're the molecules that do stuff in cells. And so DNA is the instruction to make the protein. The protein's the actual molecule that does everything that the body needs. So DNA is incredibly important because DNA holds all the instructions and on all the proteins found in our cells. So let's look at some more slides on DNA. So first of all, DNA is found in chromosomes, and we covered this in the Santhi lab. So someone asked a question, I believe, about Santhi. Let me pull up that question. Purpose of Santhi. So the real purpose of Santhi for us in biology was to give this idea that our bodies contain lots of DNA. You know, we have 46 of these things called chromosomes. And chromosomes are just our DNA all packed up in a nice little bundle. We saw them during mitosis when we need to separate them, but they're all packed up. But in reality, most of the time, the DNA is in a long strand, thread-like structure that looks like this picture here. This is our iconic double helix molecule. But not all the DNA is used all the time. DNA is used in sections called genes. And genes are made up of a certain number of these nucleotide bases. And this sequence is what codes for a specific protein. And so the gene is found in the molecule of DNA, and our DNA is found in chromosomes. So you can kind of see the hierarchy of these structures. So Santhi taught us that having a chromosome doesn't necessarily indicate you're going to make the protein. You got to have the gene that makes the protein. In that Santhi lab, we talked about the SRY gene, and that SRY gene is what causes a male to start developing as a male during development. And that SRY gene essentially turns on, gets expressed at a certain time. And so what we found out was Santhi had a Y chromosome. She had the chromosome that should have contained the gene, but that gene wasn't present. Therefore, she didn't develop like a normal male would develop because she didn't have the correct gene to make the protein to get her to turn into or develop into a male. So that's one thing you need to know about the central dogma. It's like what DNA is and how DNA compares to a gene. And then you also need to know what makes up DNA. And DNA is made up of these things called nucleotides. You have phosphates and sugars, and those make up the backbone. That makes up the sides, and I'll show a picture of DNA like right here. This is the backbone of the DNA molecule. So it's pretty boring, though. It's just phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. Not very exciting. It's like more structural, kind of gives it its shape. It holds everything together. Not very exciting. The exciting part is the nitrogen bases found in the middle, like the rungs of our ladder model. So it's these nitrogen bases that are actually the B's, the A's, the T's, and the C's that make up the code. So the actual code is found in the order of these nucleotides, and that's a very important concept. We did a lot of activities that really stressed upon the fact that the order of these nucleotides, the order of these bases are what's incredibly important when we go back to here and we make a protein. So it's really that structure of DNA, the order of nucleotides, that determines how these proteins are built. And that I was talking about transcription and translation. Transcription is a process of taking DNA messages, the A's, the C's, the T's, and the G's in DNA, and turning it into a different molecule, not changing it. The DNA stays the same. We use the DNA to make a copy, but not an exact copy, a complementary copy of RNA. What I mean by complementary, you got to know your base pairing rules, like A bonds to T, C bonds to G, and so forth. But in RNA, A bonds to U instead of T. There's no thymines in RNA, but the same rules apply. So we take this coded message in DNA and we code it in a different molecule called RNA. And the purpose of this is that, especially in eukaryotes, the RNA can leave the nucleus and go to the ribosome to be used to make a protein. And the protein, the order of these amino acids that make up proteins, like this H, this V, and this M represent amino acids, the order of a protein, or the order of amino acids in a protein, is determined by the order of the nucleotides in RNA, which is determined by the order of the nucleotides in DNA. So that's a really important concept there, that the order of nucleotides in DNA determines the order of amino acids in the protein. So this question just popped up, so I might as well answer it now. What creates the rungs of the DNA ladder? The rungs of those nitrogen bases. The nitrogen bases found in the nucleotide, these bases right here. You're not going to memorize purines versus pyrimidines. So if you look at this picture, think of this as the upright of the ladder, the upright of the other side of the ladder. This right here is a rung. This is a rung. This is a rung. You can see them pictured in this molecule here, this double helix molecule that spirals. And so A bonds to T, G bonds to C, and that's what holds the two sides of the DNA molecule together. So a question just popped up. Why does A pair with U and mRNA? What makes that change from DNA? Great question. I honestly don't know. I don't think it's really important that you know why uracil's found in RNA. It just is. There might be an answer out there. I've definitely not taken the time to research it. So that's just what we find. Sometimes we just don't know why things happen in science, and this might be one of them, though I'm not quite sure. Good question, though. Glad you're curious about that. So one thing you're going to have to be able to do in the protein synthesis central dogma section of this final is be able to look at this codon chart that's the mRNA code. So again, a codon is a group of 3 nucleotides in a messenger RNA like A, U, G, and be able to use this chart to figure out what amino acids should be put in order on a protein. So, you know, AUG, if you look at the chart, A starts here, and then you go to U, and you go to G, that codes for methionine. whereas AAC, you start right here, A, and then go to the C that codes for asparagine. So these amino acids all have a specific mRNA code that code for them. And again, this mRNA code comes from the code found in DNA. And that's incredibly important. The code of DNA, the order of nucleotides in DNA, determines the order of amino acids in a protein. All right, let me take a look at some questions. Great question. What's the difference between mRNA and tRNA? Sorry if you already explained this. Don't be sorry, I didn't already explain it. But the main difference is that mRNA is the messenger. That's why it's called mRNA. It carries the message from the nucleus to the ribosome where a protein is made. The tRNA, the T stands for transfer. Now, we didn't really focus too much on that type of RNA. Basically, it's just carrying the right amino acid, and it matches up with the mRNA. If I go to a picture of tRNA and mRNA, you'll see, let's find a good one here, like this one. How, here's the mRNA molecule up here. The tRNAs have an anticodon, GAU, which matches up with CUA. And this tRNA is carrying a specific amino acid. And that specific amino acid is put in that spot because this tRNA can bind to only that code in the mRNA. So that's why. Not that important, but I guess it's important, worth mentioning. All right, let's check for other questions. I hope my description of transcription translation has answered your question, Miriam. That transcription makes the mRNA, which is the messenger molecule, and then translation takes that messenger molecule and puts the amino acids in the correct order to make the protein. Great question, Daniel. How does DNA code eventually affect the shape of the protein? So let's go back to, I think I have a picture right here. Okay, so this piece of DNA, when it's transcribed, it makes messenger RNA. This mRNA puts these amino acids in a specific order. Now, we take this a step further. The order of amino acids determines how this chain will fold, and the folding determines the shape of the protein. Think back to that paper folding lab that we did. You matched all the diamonds to diamonds and the hearts to hearts, and that made your protein this weird figure-8 shape with a bunch of weird squiggles and stuff in it. That was all because the amino acids were put in a specific order, which allowed it to fold a certain way. That, again, that order comes from the order of nucleotides found in the DNA molecule. Hope that answers that question for you. That was a good question. Let's see any other questions. This concept is not going to be found on the Reg Bio final, and not even on, well, a little bit on the honors final, but that review session will take place tomorrow night. So this must be an honors biology student asking this. Not going to be on the regular biology final what polar versus nonpolar molecules are. Okay, building blocks of DNA and RNA, what and where they are both found in the cell. I really like the last part of this question, where are they found in the cell. So first, talk about building blocks. DNA is made up of DNA nucleotides. DNA nucleotides have a sugar, a phosphate, and there are 4 different kinds of nitrogenous bases. You could have adenine, cytosine, guanine, or thymine. Whereas RNA is also made up of nucleotides, but they're different in the sugar that they contain. RNA nucleotides contain ribose sugar, whereas DNA nucleotides contain deoxyribose sugar. And RNA has one different nitrogen base. It has uracil instead of thymine. So we have adenine, uracil, guanine, and cytosine found in RNA nucleotides. Where are they found? Great question. DNA is found in the nucleus of all cells that have one. Some cells don't, like bacteria don't have a nucleus, but in most cases we talk about cells that have a nucleus, which are called eukaryotic cells. DNA is found in a nucleus, and it's only where it's the only place it should be found. Unless the cell's dividing and the nucleus disappears, DNA is found in the nucleus of the cell. RNA, however, can be found in both places because messenger RNA has to go from the DNA in the nucleus out to the ribosome where the protein is going to be made. So let me go find a picture of transcription translation. See if we can find a good one. Yeah, this is a little complicated, actually. This is a good one. Let's take a look at this picture. Let me throw this in the slide deck. Really good question that was brought up here. So because transcription takes place in the nucleus, where the DNA is found, the DNA is not supposed to leave, messenger RNA can be found in the nucleus, but it also can leave the nucleus and go out to the ribosome where the protein is going to be made. So transcription takes place in the nucleus, translation takes place outside the nucleus. Can you explain what happens when our mRNA mutates? Well, I'm sure it happens. Messenger RNA mutates, but messenger RNA mutating is not that big of a deal because maybe only one protein will be made that's incorrect. What's more of a problem is when the DNA mutates. If you go back to this code here. If we change this code in DNA, then we change this code in RNA, and then we change the order of amino acids in the protein. So the mutation that's pretty devastating is one that happens in DNA and not in RNA. So I wouldn't really worry about what happens to a mutation in mRNA because it doesn't really cause a problem at all. All right, let's see, any other questions? You are correct, Jordan. Those are the nucleotides or the nitrogen bases in DNA, and those are the nitrogen bases in RNA. Okay, going back to this lab or question, the purpose of the DNA extraction lab was to see that DNA didn't really give us what color strawberries were. You know, we found out that strawberries we know that they were red because they reflect red light based on our spec lab, but we saw the DNA was not red. And so it tells us that DNA is really, people think DNA gives us our traits, but yeah, that's kind of true. DNA is the code that gives us our traits. What actually was red in the strawberry lab was red proteins found in the strawberry cells. Those red proteins were created because of the code found in DNA. And the Armstrong lab was really a lab to help us, one, better write CER, claim evidence reasoning statements, but also use gel electrophoresis principles and use spectrophotometry principles to figure out how much protein was in Armstrong's samples. All right, any more DNA protein synthesis questions before I move on to gene expression? I know there's been some questions. Someone just popped a question about pGlow, so we'll talk about that. All right, gene expression, genetic engineering. So gene expression. So first of all, for the regular biology final, you're not going to have to memorize all the parts of this thing called an operon. But you're going to have to know the basics as kind of how it operates. In fact, that's where the word operon comes from. So you should know that there's a gene that actually codes for something. In our lab, pGlow lab, it coded for a green fluorescent protein. There's a gene found on this operon. And there's this thing called the repressor that is like the on-off switch. So in this example, this repressor is blocking this gene, which would mean the bacteria can't read the gene, can't transcribe it, which means it can't then translate it. It can't make a protein, all because this repressor is blocking this thing called RNA polymerase. This enzyme can't get to the gene because it's being blocked. So in our lab, it was this plate here, the plate that had bacteria growing but didn't have anything glowing. That's what told us that the gene was present. It's just that the gene wasn't turned on. There was no gene turned on, so we couldn't make the protein. Sorry, I'm having a little difficulty here. Go back in the presenter view. That's better. For some reason, the screen size got really small. Hoping that's bigger now. Okay. Great question. What removes the repressor? What removes the repressor is this molecule called an inducer. In honors, we talked about this idea of lactose that we didn't really talk about in reg biology, but in biology, we just called it sugar. And what removed the repressor was the presence of sugar in the growing medium, the agar we grew the bacteria on. The repressor causes the inducer, I'm sorry, causes the repressor to be removed, which allows RNA polymerase to read the gene. You transcribe it to mRNA. That mRNA can be used to make a protein. In this case, the protein was a green glowing protein. So going back to this question, the purpose of the Pico lab taught us two things. One, it's not, or re-emphasized that it wasn't the DNA that glowed. Because if you looked at that DNA under a UV light, it would not glow. It was the proteins produced by the bacteria that glowed. What also tells us that is that the fact that if you look at these two plates, both plates had the glowing gene inserted in the bacteria. We heat shocked both of them. We add the plasmid to both of them. They both had the gene. What glowed was the fact that in the sugar plate, the gene was turned on, and the bacteria actually made the protein to glow. Now, column chromatography, I didn't put that in the slide, so let me go find a picture of that. Glow column chromatography. That's not so great. There's a better one. No. Here's a good one. I also have a video on my YouTube channel that if you want to watch on column chromatography, kind of explains the process. So basically, column chromatography works a lot like gel electrophoresis, except when you put the protein in the column, the bigger molecules actually have an easier time traveling because they don't get stuck in these little tiny, they look like little wiffle balls. They're made out of silica. And the silica has little tiny holes, and the big ones can't fit in the holes, so they kind of slip past, and they fall out first. So if you see this picture, we got big molecules and little molecules mixed together. We put them in a column, and then the big molecules fall out first, and the small molecules fall out last. And so we can figure out that the molecules collected first are the bigger ones. The molecules collected in later fractions or later tubes fall out last. And that's the purpose of column chromatography. So the purpose, again, I think to answer your question, whoever asked that question, what's the purpose of the column chromatography lab, was to purify or get just one kind of protein in our sample. And our gel electrophoresis, our SDSPAGE gel we ran, if you look back to a gel picture, going too far here, if you have one band that matches up to the high concentration of the protein, that one band indicates you have one kind of protein in there, which indicates it's pure. It's a pure sample. If you have multiple bands in a lane, it means you have an impure sample. You have multiple kinds of proteins. So column chromatography and gel electrophoresis can be used in tandem to, one, this purifies it. This gets you the pure sample. A gel would then test to see if your sample is pure. If it is, you should see one band in your gel. So this question, so in our jellyfish lab, the sugar was the inducer. That is correct. The sugar in the plate allowed the bacteria to make the protein, which allowed the bacteria to glow. In regular biology, what triggers the inducer to take off the repressor? The sugar, if you're looking for the name of it, it was arabinose. If you're looking at how it works, when the arabinose attaches to the repressor, it changes the repressor's shape. The repressor is a protein, and its shape changes. And if its shape changes, it can't attach to the DNA, and it can't block the gene from being read. But that's a lot of detail you don't really need to know. What you really need to know is that an inducer removes the repressor, an inducer turns the gene on. So yeah, Gavin, in a sense, it is kind of opposite. In gel electrophoresis, small molecules travel faster through the gel. In chromatography, big molecules travel faster through the gel. They both separate based on size, whereas gel electrophoresis, the sample is stuck in the gel. We can't collect it in a tube and give it to somebody. Chromatography, we can actually collect samples of it. So if you think about collecting the samples, we actually had a pure sample of green glowing protein in that little tiny test tube. You know, if that was an important medicine like insulin, we got the bacteria to make, then we've got a little tiny test tube of insulin. It's no use to us if we have insulin stuck in a gel. We can't give that gel to a person and say, here's your insulin for your diabetes treatment. No, we need to purify insulin, and that's what column chromatography is used for. Great question. Okay, purpose of the GMO lab and synthetic biolabs. We'll get to synthetic bio. The synthetic biolab was just kind of more emphasis that we can genetically modify organisms to make certain things. In our synthetic biolab, we kind of challenged you to figure out a problem that a synthetic biologist could solve using engineering of a living organism. So getting a bacteria, for example, to produce insulin is an example of synthetic biology. These bacteria don't normally make insulin. But if we can engineer them to make human insulin, then they can make a protein that's important for us that we can use to solve a problem. The GMO lab was to really test your understanding of, you know, if a gene was present or not in this GMO organism. So we were using gel electrophoresis to see if a gene was present. And we used PCR to see if a gene was present or not. So that's what the purpose of the GMO lab was. All right, let me see if there's any other questions. If you see a question I haven't answered yet on the list of questions people are asking, give it a thumbs up. The more thumbs up it gets, the higher on the list it gets on my end, which allows me to see it better. It stands out to me that question hasn't been answered. So again, if you see a question that that hasn't been answered and someone asked it, give it a thumbs up. The more thumbs up it has, the higher on my list of questions it comes up. If a question's already been answered a lot, you can give it a thumbs down. You know, not that it's a bad question, but it's just a question I've already answered that I don't need to answer again. So next slide, I have, you know, why do cells control gene expression? Why are these mechanisms in place? Well, a couple of reasons. So it goes back to the idea of protein synthesis, central dogma. DNA codes for RNA, codes for the order of amino acids, which fold and make a protein. This costs the cell energy. This process costs the cell resources. So a cell is only going to make a protein when it needs the protein. It's a waste of energy and a waste of resources if you're making a protein that you don't need. And so many cells control many of the genes they have to not waste energy and waste resources, so they don't make unnecessary proteins. Another reason why cells control gene expression is linking to our most recent lab, the stem cell lab, that stem cells contain all the genes, all the information needed for any cell in our body. For blood cells, liver cells, muscle cells, skin cells, nerve cells, fat cells, signaling cells, every kind of cell we have. Stem cells have the DNA for that cell. The genes just aren't turned on yet. And so when a stem cell goes through what's called differentiation, certain genes are turned on to give it the proteins to make it a blood cell, or to give it proteins to make it a nerve cell, or give it the right proteins to make it a muscle cell. And so in these specialized cells, those genes that aren't necessary, say to be a nerve cell, are turned off. They're not being expressed. They're there, they're just turned off. And what scientists are trying to do is, well, we know the DNA is there. How do we get those genes to be turned back on? How can we take a nerve cell, turn it into a stem cell, and then change it to a blood cell with the right type of chemicals to turn the genes on that we need to turn on? And that would be a huge breakthrough in medical science. So why do cells control gene expression? One, so they don't waste resources and waste energy. And then two, so they can become the right kind of cell, produce the right kind of proteins to be the cell that they are. All right, any questions about gene expression and GMOs? I guess GMOs is part of this unit. Oh, how is the process of gene expression used in making genetically modified organisms? Great question. Well, genetically modified organisms have genes that aren't normally in that organism. And so not only do scientists have to understand how to get the gene into the organism, they have to understand how to get that gene to be turned on so that modified organism can make whatever protein we want it to make. It could be a plant that's producing, more nutrition in its leaves or stems or fruits or whatever part of the plant we eat. It could be a cow that produces more milk or healthier milk. It could be a bacteria producing a chemical that we need to solve some kind of problem like human insulin. All comes down to, can we get that cell to turn the gene on and make the protein we want it to make? That's how gene expression is linked to GMOs. Can stem cells go from one function then be changed into another? The simple answer is yes. It has been shown that not in all cases that we can take certain cells, like it's been shown that we could take a skin cell and turn it back into a pluripotent stem cell. Don't need to worry about that word pluripotent, but a stem cell that has the ability to turn into other cells. It's not a perfected practice yet, but it definitely has potential that we can take stem cells that have turned into one cell, and then we can kind of go backwards in the process. This person asked, explain STEM labs. I'm assuming you mean stem cell labs. Basically, the stem cell lab showed us the difference between embryonic stem cells and adult stem cells. That embryonic stem cells have more potential. They're more versatile. They can turn into different kinds of cells, whereas adult stem cells, which are found in all mature organisms, by mature I mean organisms that have fully developed and have been born, have stem cells that can only turn into a certain kind of cell. Like your liver has stem cells that can turn into new liver cells, but those adult stem cells in your liver cannot be used to make more brain cells. So that was what that lab was for. All right, I think that covers all the questions I've seen in terms of gene expression. So I'm going to move on. The next slide. Ooh, we actually have some questions. Cell cycle, cell membrane, cell transfer. What do the spindle fibers do, and what happens if they break? We'll go over that question. Why must the cell eventually divide? What problems does arise of nutrients and waste in cell growth? So let's talk about the cell cycle first. So in the cell cycle, we have two main stages. We have the interphase stage, which takes up the majority of the cell's life. Majority of the cell's life is spent in this big interphase part, which consists of G1, S, and G2. Mitosis, which is the exciting phase when the cell actually divides, is a very short part of its life cycle. And in mitosis, we looked at, prophase, metaphase, anaphase, telophase, and that is where we get to those spindle fibers. So let's take a look. Let me go find a picture of mitosis. And here's a good one. Talk about mitosis and that spindle fiber question you had. Oops. Oh, that's a blurry picture. This one looks better. Yeah, that's good. So when you think of mitosis, first of all, please do not spend any time memorizing the steps of mitosis. But a good question that was asked is what's important about those spindle fibers, and those are important. So we take a cell, we call it the mother cell, that has its chromosomes. This one happens to have 2 chromosomes, and they get duplicated. That occurs during the S phase of the cell cycle. So during the S phase of the cell cycle, the cell says, okay, time for me to divide, got to copy my DNA. Because the two new cells that I'm going to make, that are the result of this, each need a copy. And so we replicate it. And so when we see these X-shaped chromosomes. And then the chromosomes line up in the middle of the cell. They are pulled there by the spindle fibers. So if those spindle fibers weren't there, we could not move these chromosomes into the right orientation, the right spot, to get them to separate during cell division to get 2 copies of those chromosomes in the new cells. And so without those spindle fibers, the cell couldn't divide. Or the cell would have too many chromosomes in one cell because these chromosomes might all go down here. This cell would be 4 chromosomes. This cell would have none. Those spindle fibers are really important for actually moving and separating the chromosomes during mitosis. So that was a really good question that someone put in the slide deck. Now, I saw this question pop up. Let me go find it. This is a great one. By looking at a photo of cell division, how is it possible to tell the rapid cell division is taking place? You're welcome. I'll definitely answer that question. So when you look at a photo of cells during cell division, I think this was a picture in the study guide, if I remember correctly. So this picture here. So let me look at this picture here. Let's see if I can copy that and put that in the slide deck. Blow that up. And let's view it. So normal cell division would indicate that the cell is taking its time during these 3 phases, the G1, S, and G2 phase. If a cell is not taking its time, if it's not going through the normal cell division process, it's dividing too fast, you would see more cells in mitosis if you look underneath the microscope. Right now, most of the cells in this picture are in interphase. You have interphase, 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 interphase. Most of them are interphase. Some of them are in mitosis, but most are in interphase. Most of the time, the cell is going to be spending its time doing what it's supposed to do, and that's being a cell that produces proteins, that does a job in the body. That's all happening in G1. It's not until it gets to a certain point in G1, a certain checkpoint, where it checks to see if it's big enough, if it's got the right growth factors to replicate its DNA. When it does replicate its DNA, it then goes into G2. It checks, did I replicate my DNA okay? Is my DNA damaged? Is my DNA mutated? If it's not, it goes into mitosis and divides the DNA into two separate cells, and then goes back to G1, goes back to doing its job. Cancer develops when these checkpoints are ignored. We talked about the BRCA gene as one of those proteins that functions at a checkpoint, checking for DNA damage. If the checkpoint is ignored, then the cell doesn't check to see if DNA is damaged, and it goes right into right into G2, and goes right into mitosis. Now, more mutations could result that makes this whole process go faster. That's when you develop cancer. So tumor suppressor genes, which we'll get into, control these checkpoints. They slow the cell down if there's damage to the DNA. If those tumor suppressor genes go bad, there's no way to stop the cell cycle from speeding up if a mutation causes it to go too fast. And that would look like all the cells would be in mitosis. You would have barely any cells in the interphase. They'd all be dividing rapidly. It would be very hard to take a picture of a cell in interphase because it doesn't stay in interphase very long if the cell is cancerous. So that's a great question, going back to that one. So how is it possible? How is it possible to tell if rapid cell division is taking place if there are lots of cells that are going through mitosis? All right, let's move on to the next slide. That was on the cell membrane. So that kind of talked about, someone's talked about the different checkpoints. Sure, I can talk about those again. Can we go over the different checkpoints? Yes, let's go back to this slide here. And so checkpoints, we have three major checkpoints. We have the G1 checkpoint, checks for nutrients. Is the cell healthy? Has the cell been growing? And is there damage to the DNA? If no, all those things are okay. Goes to the S phase, copies its DNA. Then we go to G2 checkpoint. Is the cell still big enough? And has the DNA been replicated properly? If yes, we go into mitosis, and then we check if the chromosomes lined up correctly. So right in mitosis, basically have these chromosomes lined up correctly so we can divide them. Basically, we're checking to see if mitosis will happen correctly in that mitosis or the cell division checkpoint. So those are the three checkpoints that are important for the cell cycle. All right, I'm scrolling through to see if I see any more questions on cell cycle. Okay, here's a good question. What stage do the nucleus and cytoplasm divide? The nucleus divides during mitosis. Cytoplasm divides during cytokinesis. Those are really two separate things. They both happen during the division part of the cell. So if I go back to this picture, this picture is kind of a bad representation of that because it looks like it's happening at the same time. But in between this and this, mitosis separates these pieces of DNA into two separate piles, basically. And then cytokinesis is what creates two new cells. So the stage, they both happen during the division stage, but they're two separate things that happen. There's two separate processes. Nucleus divides during mitosis, cytoplasm divides during cytokinesis. I kind of already went over this, so I hope you were listening to that part. If not, you can watch this video again. It'll be on my YouTube channel. You can rewind it and find that part, the importance of G1, G2, and mitosis checkpoints. Okay, this person asked the purpose of the BRCA lab. The BRCA lab was to really show us that the BRCA gene doesn't cause cancer. It helps prevent cancer. That the tumor lane in that lab showed no BRCA gene present. That the BRCA gene was actually mutated, and we didn't have the correct gene to stop a tumor from forming. I'll get into the sea urchin lab in a little bit. Okay, this person asks, what's a lipid? The building blocks and function. I'm going to go over it. We don't really talk about the building blocks lipids in Reg Bio, so I'm not going to really go over that. But the function that we focused on is the lipids create the cell membrane. These little tiny gray, tannish colored structures of these phospholipids that make up the majority of the cell membrane. And then there are some proteins, these big purple things, sprinkled in. And so the lipid layer, it's actually a bilayer. See one layer down here and the next layer, the upper layer up here, create the boundary between the outside world of the cell and the inside world of the cell. So it creates that separation between what's the cell and what's the environment. And because this is made of lipids, these lipids always move around, which allow things to move through them. And that's what got us into cell transport. So one of the things we talked about was the fact that when a cell gets really big, diffusion takes too long. And so one reason that cells stay small and divide is because diffusion takes too long in a really big cell. And that's kind of an important concept. Another reason why cells divide is for replacement of dead or damaged cells, for growth, but also cells divide to stay small. Now, we talked about 3 different cases of cell transport in terms of water. We talked about water moving into cells, and that is when there's a higher amount of water in the environment compared to inside the cell. You don't have to memorize the word hypotonic, but hypotonic means there's less solute outside than inside. So because there's less solute outside, there's more water outside. Water moves from high to low. It's a special case of diffusion we call osmosis. All diffusion is going from high to low until equilibrium is reached. Equilibrium means things are the same. The opposite situation, when you have less water outside, more water inside the cell, water will leave the cells. We call this hypertonic. There's actually more solute, more dissolved stuff in the environment than there is inside the cell. So there's less water out here, so water goes from high to low and moves out. The cell shrinks. This happens until the cell reaches equilibrium, where it's in an isotonic situation, where it's equal. The inside and outside are equal, therefore the cell does not change shape anymore. So if you place a cell in an environment that has the same concentration outside as it does inside, the cell won't change shape. And we saw that in our potato/carrot lab. There was some cells that barely changed any mass, didn't change in mass because they were in an isotonic environment. So all cells try to reach this isotonic equilibrium state. They try to become equal because those water molecules can move across the cell membrane. All right, I'm going to go scroll through here and answer any last bit of questions. I didn't really cover cells transport all that much because that's the most recent unit, so hopefully that's a little bit more fresh in your minds. I see some questions here. How do stem cells become differentiated or specialized? Through gene expression, through the turning on and turning off of certain genes. Okay, this question, does a teenager have embryonic stem cells? No, because you're not an embryo. Once you're a teenager, once you're born, once you're at a certain stage of development before you're born, all your embryonic stem cells have gotten jobs, have gotten instructions on how to be all the different kinds of cells in your body. We do have adult stem cells, though. You do have cells that can replace, for example, your liver has stem cells to make more liver cells if it needs it. Not all organs have stem cells, so it's not a perfect situation, but your body does have some adult stem cells. They're just not as versatile as embryonic ones. Do only chromosomes divide in mitosis? Technically, yes, that's the definition of mitosis, dividing the DNA. But if you're thinking about cell division, everything else about the cell divides as well. So all the organelles that we haven't really covered, they're divvied up into both cells too. All right, looking through more questions. I think I've covered almost everything. All right, so this will be the last question I answer unless another one pops up in the next few minutes. Was the final part multiple choice and part written? What is the format? It is roughly around between 40 and 50 multiple choice questions, and there's going to be probably around 2 or 3 written questions. So it's about the same format as all of the other tests you have taken, just a little bit longer, because 1, it's a final, and 2, you've got 90 minutes to take it. So that's going to be the format of the final exam. Why must cells eventually divide? Problem arises with nutrients waste as the cell grows. Oh, and I saw that question. I never answered it. Cells divide because if it's too hard to get nutrients if the cell is too big, because diffusion is too slow. It's also hard to get rid of waste if the cell is too big, because the cell can't get rid of the waste because diffusion is too slow. So cells divide to stay small so they can get nutrients faster and they can get waste out faster. Waste being like carbon dioxide or other molecules that the cell don't need. Purpose of sea urchin lab, exclamation point. Really want to know the answer to that, huh? Purpose of the sea urchin lab, 1, it was just really stinking cool to see living cells actually divide underneath the microscope. And also the sea urchin lab emphasized or brought up the idea of stem cells, that these cells we saw underneath the microscope didn't have jobs yet. They were stem cells. They were cells that didn't have a functional role, but if we would have left those embryos continue to develop and nurture them and keep them in the right environment, they could have differentiated and turned into all the different kinds of cells required to make a new sea urchin. So the purpose of the sea urchin lab was to kick off our understanding of stem cells and their importance of stem cells. Really cool lab, though, seeing those sea urchins. Okay, I think I just answered this. It's going to be multiple choice, about 40 or some questions, and then written questions as well. All right. Scrolling through the questions, I think I've covered everything. If I'm missing a question, I apologize, but it is 9:13. Purpose of carrot lab, I'll do that real quick. Purpose of carrot lab was to understand when a cell gets bigger, it's gaining water through osmosis. When a cell gets smaller, it's losing water. And so we were able to discover this process right here, this idea of cells getting bigger when they're in a low concentration solution. Excuse me, and cells get smaller when they're in a high concentration solution. So like in our lab, the 0% solution, the carrot gained mass. In the 20% sugar solution, the carrots lost mass. That was the purpose of that lab. All right, so it's 9:14. This is going to be Mr. Wolf signing off. Thank you to all that participated and asked a lot of great questions. Best of luck to you all in your further studying for the final, and I will see many of you tomorrow. Have a good night.